Ctenophora is a phylum of animals that live in marine waters worldwide. Their most distinctive feature is the combs groups of cilia which they use for swimming. They are the largest animals that swim by means of cilia. Adults of various species range from a few millimeters to 1.5 meters in size. Like cnidarians, their bodies consist of a mass of jelly, with one layer of cells on the outside and another lining the internal cavity. In ctenophores, these layers are 2 cells deep, while those in cnidarians are only 1 cell deep. Some authors combine ctenophores and cnidarians in one phylum, Coelenterata, as both groups rely on water flow through the body cavity for both digestion and respiration. Increasing awareness of the differences persuaded more recent authors to classify them as separate phyla. Ctenophores also resemble cnidarians in having a decentralized nerve net rather than a brain. Genomic studies have suggested that the neurons of Ctenophora, which differ in many ways from other animal neurons, evolved independently from those of the other animals. Almost all ctenophores are predators, taking prey ranging from microscopic larvae and rotifers to the adults of small crustaceans. The exceptions are juveniles of 2 species, which live as parasites on the salps on which adults of their species feed. In favorable circumstances, ctenophores can eat 10 times their own weight in a day. Only 100 to 150 species have been validated, and possibly another 25 have not been fully described and named. The textbook examples are cydipods with egg-shaped bodies and a pair of retractable tentacles fringed with tentilla that are covered with chloroblasts, sticky cells that capture prey. The phylum has a wide range of body forms, including the flattened, deep-sea platyctenids, in which the adults of most species lack combs, and the coastal baroids, which lack tentacles and prey on other ctenophores by using huge mouths armed with groups of large, stiffened cilia that act as teeth. These variations enable different species to build huge populations in the same area, because they specialize in different types of prey, which they capture by as wide a range of methods as spiders use. Most species are hermaphrodites. A single animal can produce both eggs and sperm, meaning it can fertilize its own egg, not needing a mate. Some are simultaneous hermaphrodites, which can produce both eggs and sperm at the same time. Others are sequential hermaphrodites, in which the eggs and sperm mature at different times. Fertilization is generally external, although platyctenids' eggs are fertilized inside their parents' bodies and kept there until they hatch. The young are generally planktonic and in most species look like miniature cydipods, gradually changing into their adult shapes as they grow. The exceptions are the baroids, whose young are miniature baroids with large mouths and no tentacles, and the platyctenids. In at least some species, juveniles are capable of reproduction before reaching the adult size and shape. The combination of hermaphroditism and early reproduction enables small populations to grow at an explosive rate. Ctenophores may be abundant during the summer months in some coastal locations, but in other places they are uncommon and difficult to find. In bays where they occur in very high numbers, predation by ctenophores may control the populations of small zooplanktonic organisms such as copepods, which might otherwise wipe out the phytoplankton, which are a vital part of marine food chains. One ctenophore, Nemiopsis, has accidentally been introduced into the Black Sea, where it is blamed for causing fish stocks to collapse by eating both fish larvae and organisms that would otherwise have fed the fish. The situation was aggravated by other factors, such as overfishing and long-term environmental changes that promoted the growth of the Nemiopsis population. The later accidental introduction of phērō helped to mitigate the problem, as phērō preys on other ctenophores. Despite their soft gelatinous bodies, fossils thought to represent ctenophores, apparently with no tentacles but many more comb rows than modern forms, have been found in Lagerstätten as far back as the early Cambrian, about 515 million years ago. The position of the ctenophores in the evolutionary family tree of animals has long been debated, and the majority view at present, based on molecular phylogenetics, is that cnidarians and bilaterians are more closely related to each other than either is to ctenophores. A recent molecular phylogenetics analysis concluded that the common ancestor of all modern ctenophores was cydipod-like, and that all the modern groups appeared relatively recently, probably after the Cretaceous-Paleogene extinction event 66 million years ago. Evidence accumulating since the 1980s indicates that the cydipods are not monophyletic. In other words, do not include all and only the descendants of a single common ancestor, because all the other traditional ctenophore groups are descendants of various cydipods. Distinguishing features: ctenophores form an animal phylum that is more complex than sponges, about as complex as cnidarians, and less complex than bilaterians. Unlike sponges, both ctenophores and cnidarians have cells bound by intercell connections and carpet-like basement membranes, muscles, nervous systems, and some have sensory organs. Ctenophores are distinguished from all other animals by having colloblasts that capture prey by squirting glue on them, although a few ctenophore species lack them. Like sponges and cnidarians, ctenophores have two main layers of cells that sandwich a middle layer of jelly-like material, which is called the mesoglea in cnidarians and ctenophores. More complex animals have three main cell layers and no intermediate jelly-like layer. Hence ctenophores and cnidarians have traditionally been labeled diploblastic, along with sponges. Both ctenophores and cnidarians have a type of muscle that, in more complex animals, arises from the middle cell layer, and as a result some recent textbooks classify ctenophores as triploblastic, while others still regard them as diploblastic, ranging from about 1 mm to 1.5 meters in size. Ctenophores are the largest non-colonial animals that use cilia as their main method of locomotion. Most species have 8 strips, called comb rows, that run the length of their bodies and bear comb-like bands of cilia, called cteines, stacked along the comb rows so that where the cilia meet, those of each comb touch the comb below. The name Ctenophora means comb-bearing, from the Greek kappa tau epsilon iota sigma meaning comb, and the Greek suffix phi omicron rho omicron sigma meaning carrying.